So I'm going to talk a little about what I learned at the public lecture by Alec Askimov from the University of Illinois at Urbana. Um, it's on the physics of genes and the promise of personalized medicine. And I'm going to talk a little bit about, the projects that they're working on, the research they're working on, and how they are trying to find personalized medicine in the future. So the first thing I talked about was DNA and really the structure of DNA and a little bit of the characteristics of DNA because that's very important to study and to know if you are trying to get into a cell and treat it with a specific medicine. So DNA has a highly negative charge and it's about 2 nanometers in diameter. In 1990, the Human Genome Project was launched by the US government, and that was used to determine the sequence of nucleotides, base pairs that make up DNA. And so that was a really, really big project that actually finished early on, and it was a very successful project, and it is now used to help for molecular medicine, which is really important for genotyping of viruses and things of that sort. So DNA in a medium, the same side charges will attract, which is what happens, so DNA is surrounded by a lot of counter ions, and which causes like DNA molecules in NaCl to repel, but they will attract in magnesium chloride, which is a very interesting concept. So it's pretty much where half of a charge will interact with one DNA and such. So beyond the DNA sequence, epigenetic regulation is something that Alec talked about a little bit. It is able to control DNA condensation. Also, DNA molecules unite by sharing polyamine, and the more sharing, the more attraction there is. Some abnormal operation of protein motors that process info in DNA are causes of genetic and multifactorial diseases, which is what cancer happens to fall under. So building a computational model of the repliosome is something that Alec and the team is working on. They are working on, it's pretty much replicating other DNA and getting that DNA into the cell and like repairing it or replacing it and that kind of thing. It's so they're trying to build a computational model of that, and it would have all the essential components. And then DNA repair, pretty obvious, it's essential to surviving. So that's something that they're trying to do. There is great potential of nanopore. Nanopores at first weren't very popular, but they're becoming more popular now, and they can be really good for detection and manipulation of single biomolecules. And so that can be moved to like silicon nanotechnology and then also quantum sequencing of DNA. To wrap things up, Alec talked a little bit about the next step that the team is trying to make, and they're trying to target disease cells, which is obviously very important. You don't want to be going into every single cell if you're just trying to treat specific cells. They're trying to prevent degradation before reaching that target, which can be very hard within the human body. There's a lot of different things going on, and trying to reach that cell can be difficult without the medicine, the DNA replication, the replisomes, everything like that changing before it reaches the target. And then getting the drug into the cell is another really big thing due to the nature of a cell and getting things into it. So there was in 2009, I believe, a DNA origami box, which was a cancer-killing DNA robot. And it's something you can look up online. It's very interesting how it will go in and would kill the DNA like in a DNA cell or in a cancer cell. And Alec actually talked a little bit about a DNA syringe, which DNA origami syringe, which would actually like latch onto the cell and then be more of a syringe and get into the cell that way. So that's something that they're looking at. So essentially, DNA sequencing will become a routine diagnostic tool It's becoming more and more popular, and it's also becoming cheaper, which is really cool. Makes it more useful, especially in research and everything like that. So yeah, overall, I thought the lecture was very interesting, and I didn't realize how much biology and physics overlap to, be able to do things like this. And who knows, eventually we'll cure diseases and cancer cells that we had never before had the answer.